Well, thanks very much, David, and thanks for the invite to be here. And I'm just delighted to have this opportunity, actually with my mask on during the breaks. We'll be interacting with many of you. You know, it's sobering to think that I've been director now for about 2.5 years, and I was hired by Zoom. I've been interacting with most of my colleagues across the Institute by Zoom, interacting with many of you by Zoom. And while I've come to appreciate how powerful Zoom is and how much work you can get done by Zoom, actually it's just not the same as being in person. So I'm delighted to be here. I look forward to doing a lot of listening during the Exposome meeting today and tomorrow, as well as during the Course Center meeting at the rest of the week. So anyway, it's again, just great to be here. So what I want to do is just take a few minutes and build on some of the comments that David just made, and also tell you about some of the workshops that will be happening over the course of the next several weeks. So let me just start off here, and I'm going to borrow a diagram from, I guess Gary and Roel are in the audience here, but I use this often. And the way I use this diagram is to illustrate the enormity of the challenge that we have within the environmental health sciences community. I mean, we've been studying many of the things in the lower compartment here. We've got to do a lot of work on pesticides and POPs and plastics. Actually, we'll probably be doing more on plastics and plasticizers over the course of the coming years. I mean, water contaminants, air pollution, lots of different things here. And for the most part, we've been doing this one at a time because that's what the technologies that we have allowed us to do. But truly, if in the environmental health sciences community, we don't want to know just about pesticides or just about PM 2.5. We want to understand the role of the environment on human health. Actually, are there good things in the environment or potentially bad things in the environment that can cause adverse health effects? So what it means is that we need this new paradigm. So it's not just about studying one of these things, it's about studying all of these things. But most importantly, if we want to understand the environment, that's a big part of what we're going to be talking about over the course of the next day and a half, is we've got to be factoring in all of the social, the ecosystems, and the lifestyle variables into studying the environment. Now, as David indicated, back in 2005, Chris Wilde here proposed the concept of the exposome, which was terrific. I think most of us embraced the exposome. It was about studying all these different things because the exposome is the research framework that allows us to take all of these things into consideration to evaluate the impact of the environment on human health. But the problem is, how do you do an exposome experiment? In fact, when I was interviewing for this job, Dr. Collins, who was the director of the NIH at the time, we were talking about a variety of different things, and I talked about how excited I was about the whole new framework of exposomics. He said, well, Rick, he said, I think you're getting many of us pretty excited about this, but how do you do an exposome experiment? Hey, I'm interested, he's saying, in my lab, I want to do an exposome experiment. Josh Gordon, the director of NIMH, is asking, look, Rick, I want to do an exposome experiment. How do I do it? And actually, Walter Koroshetz recently gave a talk at our council meeting, very interested in exposomics. But Francis brings the perspective of genomics. But genomics is pretty simple. You develop these sequencing machines, you start at one end of the genome, and you knock out nucleotides. And when you're at the other end of the genome, you're done. So how do we do this? So one of the things that we did was we actually had Gary Miller give a presentation, actually coincident with the presentation by Walter Koroshetz, who was the director of NINDS. And Gary actually proposed that we need an operational definition of the exposome. I think David was kind of getting at this point too in his introductory remarks. So we're going to need to, so how do you do an exposome experiment? I mean, it's probably going to have multiple different parts, but it's likely that we're going to need some new technologies to actually capture the totality of exposures. But we don't want to wait for all these new technologies to be developed. And I think Gary was talking about, let's start with the pragmatic exposome. Gary, I hope I'm getting that right. So focus on what we can do now. For example, high-resolution mass spectrometry is a great tool that we can use to actually capture more than one exposure at a time. So let's start with what we can do. And Gary and I were on the phone recently. We were talking about how we could really model this on the genomics community. I mean, if the Exposome is the environmental equivalent of the Genome Project, well, let's learn from what the genome scientists did. Back in the early days of genome science, they didn't have all the sophisticated technologies that we have now. They didn't have genome sequencing machines that could knock out 3 billion nucleotides for less than $1,000. The first genome took $2 billion. Not very cost effective and certainly not very high throughput. So let's model this in the genomics community over the course of time. In fact, as part of the Genome Project, new technologies emerged, and the communities embraced these. Well, they helped develop them, and the communities embraced the technologies as they emerged. So we can do the same thing. And so one thing that we're going to be doing, I always like, as director of the NIEHS, I like to make things happen. So when I've heard that there are questions about what is the Exposome, and again, David, I think you very nicely articulated this, let's have some workshops. Let's get people together. Let's brainstorm about what we're doing, and then hopefully I can come back, and when Francis Collins emails me and asks me how to do an Exposome experiment, I can tell him specifically how to do this. So under the leadership of Michelle Bennett, who is she's a special advisor to the director. She actually came from the National Cancer Institute. She has been working together with many of you in the audience, and as well as many of my colleagues across the environmental health sciences community. And you all, and together with Michelle and Alex Torres and others, have been planning a set of workshops. And these workshops are going to happen over the course of the next several weeks. So fasten your seatbelts. Actually, fasten your shoulder straps. This is going to be a wild ride over the course of the next several weeks. So what I thought I'd do is take you through this. Now, if you're interested, I hope we have the slides are available, or if you don't have the link, just get on our website and we can give you access to the, and these will be virtual. So 5 different workshops. It's hard to actually jump on an airplane and hop to different parts of the country for workshops, but you should be able to do this. And we're going to be using something called Open Space, which is a meeting technology that I actually brought to with Linda Birnbaum in our first strategic planning. It's getting people in the room together and trying to extract exciting new ideas from each person's brain as they come to these workshops. So let me take you through, just take a few minutes and talk about the 5 different workshops. In fact, it was this committee that came up with the 5 different workshops. So here they are, and these will be happening starting on July 22nd. And so let me take you through. So this is all about accelerating precision environmental health, demonstrating the value of the Exposome, as well as, and it's not just about precision environmental health. David talked a little bit about this, but how do we now operationalize the Exposomes? Even if you're not doing precision environmental health, how do you collect the data in a way that we can be all working synergistically together, but most importantly, factoring in all of the elements that are responsible for environmental impacts on human health. So the first workshop is about tools, technologies, and methodologies. So David took you through some of the technologies that we've already supported. We have questionnaires. They're GIS environmental models. Some powerful tools that are emerging by looking at GIS coordinates and then correlating that with say, concentrations of PM 2.5. It's remarkable what you can learn about environmental exposures by knowing someone's address, where they live and where they work, or where they've lived and where they worked. So powerful things that are happening. Pictures about cosmetic use, food, cleaning products. Also, a lot of work that's been done around biomarkers on different tissues, collecting different samples. And of course, we have the omics technologies, including the high-resolution mass spectrometry, as well as mobile devices, actually the bracelets, the plastic, whatever kind of bracelet they are, but absorbing all of those nasty compounds and giving you a better sense of what is it you've actually been exposed to on a personal level. So that'll be happening on July 22nd, 11 o'clock to 4 o'clock, again, using Open Space. We just ask that you register and then show up. That's part of the issue with open space. What you do is you show up and you bring your passions and your interests, and there are then systematic ways to capture your interest and those passions. The second workshop is on August 5th, and this is about the biological responses and impact in health and disease. So this, I think, is the recognition that environmental exposures actually have effects on human biology. So there may be somatic mutations that occur as a consequence of the exposure of different environmental chemicals. Or there may be epigenetic modifications that happen in response to sleep disorders or social impacts of the environment on human health. And there may be ways of looking at changes in gene expression, both at the level of messenger RNA or different non-messenger RNAs or proteomes, as well as metabolomes. So the second workshop is really about figuring out, is there a way to operationalize how individuals respond to this vast assortment of different environmental exposures. The third workshop is about the future of clinical and prevention trials, cohorts, and epidemiology. It probably more directly relates to the precision environmental health concept that David was talking about. And of course we have, I think many of you know, we have the ECHO cohort. And the ECHO cohort is using the HERE resources, and they're studying multiple exposures on mothers and children and a variety of others within their cohorts. But you have a number of other, and as new technologies emerge, we'd like to be able to seamlessly integrate those new technologies into the ECHO cohorts. But you have all these other programs, the All of Us program, 1 million-person cohort, genetically highly heterogeneous. You have the International Disease, ICDA, the International Common Disease Alliance. And this is an attempt to bring human geneticists from across the globe working collaboratively together. So there are tens, if not hundreds of millions of patients and members of these cohorts, and these genomes will be sequenced. But right now there's this sense that if we sequence someone's genomes, that's going to give us all of the information that we're ever going to need to be able to predict health outcomes. Well, guess what? People get sick not just because they've inherited sequence variants from their parents. They get sick because of environmental exposures. So we're making a lot of progress on this. So this workshop is about integrating the exposomics into the types of work that's happening with these large genetics and genomic experiments and cohorts across the globe. So I think it's going to be very insightful. And at the same time, when we do that, we're going to be able to extract from that collaboration with these human geneticists a great deal of insights into the gene by environment effects that are happening associated with environmental exposures. The other part of this, and we're going to hear a lot more about this over the course of today and tomorrow, so I'm not going to go into great detail here, but this is all about we absolutely have to integrate the social and the societal impacts of the environment into the exposomics science. So Paul and Daryl, I believe, are going to be talking a lot about this. We talked a little bit ahead of time today. This is all about the public health exposome conceptual model, and I know that I'm personally looking forward to hearing a lot more of the details. This will be the workshop that will be happening on August 19th. So Daryl, you going to plan to be there? Okay, good. Glad to hear it. Okay, so then the final thing I want to point out is that it's about data. So on August 26th, there will be a workshop from 11 to 4 o'clock, and it's about data collection. So right now we have the HERE resource that David alluded to, and this just gives you a sense of the enormity of the challenge. So just with HERE, in 46 different studies, there were, 72,137 specimens, 38,020 subjects, and the number of chemicals targeted is 139. So there's just a huge amount of data from just a limited number of studies. Now, if you expand this globally, I mean, if the exposome is actually going to work, we need to have data standards. We need to know that when we're collecting data in one place and in one country, that this can be seamlessly integrated together in data repositories that Bob can actually use for his exposomic science, to be able to blend all this together and actually achieve some of the end results that David was referring to. So that workshop, again, will be on August 26th. And then hopefully over the course of these 5 workshops, we'll be able to come out of this with a better sense of how do we do exposomics experiments? What is an exposomics experiment? And furthermore, it's what's not. I mean, not everything has to be falling under the buzzword of exposomics. There's still a lot of fabulous environmental health science that can get done outside of calling it an exposomics experiment. So I think that's it. And again, I look forward to lots of discussions.